Hi, my name is Jacob Sharpe, and I'm from Telluride High School, and I did my internship in the Harvard Medical School in the Wu Lab. So, yeah. So, right when we got to the lab, we were meted by our amazing lab manager, Mira. She wrote that nice note for us. And this is where me and Kiara, the other Pintern, sent. This is our work area. Some general lab stuff, chemicals and such. And then this is the lab garden, which Mira showed us, I think, the first day. She was very excited about this, and we went there a few times and harvested some vegetables. So that was nice. So the first real science we did was under my first mentor, Laura. She took us to the microscope right away. Smart move by her. Bring us the interesting stuff. But basically, we had these cells that she cured, and we looked at them. They had these protein marks for G1 or G2 phase of the cell cycle, and we could do some analysis over here to see which part of the cell cycle they were in. After this, I moved into some cell painting. Basically, we dyed these cells with 5 different dyes, cured these ourselves, did some wet lab work. And from this, you can see different parts of the cell. So that was very cool. We dyed that. And then I moved into some more real research under my other mentor, Faye. So Faye was researching telomeres, which are these little caps on the other chromosomes. They're a repeating DNA sequence, and they're directly linked to aging. So that was her main focus. The picture on the right, picture on the right is the telomeres, and then this is the DOPI, which is just DNA. And she asked us to go home and come up with a question. So my question was, do telomeres localize to euchromatin or heterochromatin zones? I did not know what that means at first, but
But it's clear to all of us.
yeah.
Super clear.
Basically, euchromatin is less dense areas in the nucleus where it's more transcriptionally active, and heterochromatin zones are more dense, less active. So I had to do a bunch of stuff to answer this question. A lot of coding, a lot of data analysis from the pictures Faye gave us. I actually didn't write any code myself. It was all ChatGPT. It's pretty good at it these days. But yeah, it gave me great code. I was able to do everything. And from this, I got some statistics. This is pretty boring stuff, but basically, we rejected the null hypothesis and concluded that the distribution is not random, and it leans towards the heterochromatin zones. So the telomeres like to be in the less transcriptionally active zones, which makes sense for a few reasons. Telomeres prefer to associate with repressive areas because they don't want to be seen as damage. This may have implications to telomere function and stability, and then also it supports the telomere position effect over long distance theory, which says that telomeres affect other genes around them, which is just a theory, but that's what Faye is researching currently, or part of her research. And so this is where we lived. This was our house, which Kiara went into. That map on the right is where we lived in Alston. If you went down this street, you got to BU. If you went down this street, you got to BC. We had a park right. It was a great place to live. I think a 15 to 20 minute bike ride from where I worked. So there was a lot of cool stuff around too, a lot of good restaurants. It's a very nice place to be in Boston. Outside of the lab, I tried to spend as much time as I could outside, just exploring. I went to every college campus, Harvard, MIT, BC, BU. I went to the Arboretum, which I guess Harvard just owns a forest. That was pretty cool. Yeah, they got a lot of money. And then there was the aquarium, which I really liked. They have a big turtle there named Myrtle. Pretty cool turtle. And then there's also the Boston Commons, which was a bike ride away, so I went there a lot to see some nature, which I didn't get as much of in Boston as I did here. One thing I want to mention is the museums. So our amazing lab manager, Mira, took us to every museum in Boston, I think, or almost all of them. She took us to the Museum of Fine Arts, Museum of Science, Museum of Natural History, and yeah, she made sure to take a picture of me with every globe in the Museum of Science. Yeah, there's like 10 pictures, a lot more globes. But yeah, I just thought I should mention that. We had a great lab manager. Me and Kiara were very lucky. And advice for future Pinterns. So I know everyone says this, but ask a lot of questions. If you don't know what's going on your cook, like, you gotta just ask. You gotta ask. And then get excited about your research. I got really into my research question, partly because my mentor was super excited about, but also it's just interesting stuff. And if you get into your research, it'll be a lot more fun. And then get to know your mentors and coworkers. Like, eat lunch with them, try to hang out with them. They're really cool people, and they'll be good to know in the future, I think. And then also, spend as much time as you can exploring. Like, it's easy to just sit in your house and play on your phone or cook or something, but you should really go out. Where everyone was is super cool, interesting places, so explore it. Use your time. And thank you to all these people.
Awesome.